Hello, everyone, and welcome to today's webinar. My name is Lisa, and I'll be your moderator. I'm so glad you could join us. Today, we'll be exploring the power of shear flow systems with a lab demo and discussion led by our speaker, Stephanie Proano. Just a couple of quick housekeeping items before we begin. This webinar is being recorded, and we'll share the link with you afterward so you can revisit the session or share it with colleagues. We encourage you to ask questions at any time using the Q&A box. We'll collect them and address as many as we can at the end of the presentation. And to make the most use of today's time, we have pre-recorded some sections of the demo for you. And now it's my pleasure to introduce our speaker. Stephanie Proano is an application scientist with extensive experience in leveraging shear flow systems to study cell behavior, adhesion, platelet aggregation, and antimicrobial responses. Stephanie, thank you for being here with us today, and I'll hand things over to you.
Welcome, and thank you for joining today's webinar. Today, we'll demonstrate an experiment that models immune cell recruitment to an inflammation site under shear flow using the bioflux. Specifically, we'll track the adhesion of Jurkat cells, a human T cell line, to a TNF-alpha activated monolayer of cardiovascular endothelial cells that is undergoing a state of inflammation. Before heading into the lab, I'll give you a quick overview of the bioflux, our shear flow technology that enables physiologically relevant experiments at high throughput. For those of you who have joined the demo or are watching this video at a later time, you've likely considered how shear flow impacts biological processes like cardiovascular and immune system function, cell morphology, behavior, and gene expression, tissue organization, and drug delivery and treatment efficacy. And within the microbiology space, shear flow also plays a role in the establishment and progression of biofilms within the context of antimicrobial resistance, food safety, medical device-related infections, and host microbe interactions. But applying shear flow can be tricky because there is often a trade-off between designing reliable and reproducible physiological experiments where the user has full control of shear flow delivery and throughput. Static assays, for example, are high throughput but fall short on physiological relevance. On the flip side, laminar flow cells and DIY systems can mimic real conditions that can't scale easily and face serious limitations when it comes to shear flow control and the generation of reliable and reproducible results. This reality was reflected in a survey that we conducted asking scientists whether they use shear flow, how they're using it, and where challenges arise. When asked whether they use shear flow, most respondents reported that they either rely on a DIY system or that they would like to incorporate shear flow into their experiments. And common limitations with current setups include poor shear flow control and the inability to conduct reliable and reproducible experiments at scale. Without a dependable method that can generate enough replicates, it is hard to draw solid conclusions about biological processes under flow. That's where the bioflux platform comes in. It combines pneumatics, microfluidics, and live imaging to deliver precise shear flow to up to 200 dynes per centimeter square in a low maintenance format and at high throughput, while offering temperature control and gas regulation to facilitate the maintenance of physiological conditions. The system includes the bioflux controller, plate interface, and bioflux plates, which deliver air pressure to drive shear flow, an inverted microscope with a high-speed camera for real-time imaging, and an automated stage with a well-plate adapter to support image acquisition at high throughput. All hardware components are controlled through the bioflux and Montage Plus software, which streamline flow delivery, image acquisition, and data analysis. The mechanics of the system are fairly simple. The bioflux plates are placed on an inverted microscope to acquire live images of experiments. The bioflux controller pushes air or a custom gas through tubing to move fluids from the wells of plates into the microfluidic channels of bioflux plates. The bioflux offers the ability to generate pulsatile, laminar, oscillatory, and bidirectional flow, all while keeping your samples away from the tubing but contained within bioflux plates, which consist of PDMS microfluidic flow cells that are embedded within standard SBS well plates in a 170 micron glass cover slip, perfect for high-resolution imaging. The microfluidic design has viewing windows on the bottom of the plate where experiments take place and are imaged in real time. The plates are available in 4 formats that vary in their capabilities and architecture. The 48-well plates have 24 microfluidic flow cells. They can either be low shear, delivering up to 20 dynes per centimeter square, and high shear, delivering up to 200 dynes per centimeter square. The microfluidic flow cells consist of one inlet well that is connected to one outlet well. Each viewing window within the plate accommodates 2 microfluidic flow cells that are independent of each other, where both the experiment and image acquisition happen. The 24-well plate has 8 microfluidic flow cells and can deliver up to 20 dynes per centimeter square. Because this plate has 2 inlet wells that are connected to one outlet well, 2 different fluids can flow within the same microfluidic flow cell to generate a parallel flow stream, enabling the creation of physical and temporal gradients. Last but not least, the 6-well plate has 3 microfluidic flow cells and can deliver up to 60 dynes per centimeter square. We currently offer 2 models of the BioFlux system. The BioFlux 200 Plus, which features temperature control, 2-phase and pulsatile flow. It can work with 2 plate formats, the 24 and 48-well plates. It has the ability to conduct up to 24 assays per plate and adapts to an existing inverted microscope. The other model we offer is the BioFlux 1000 HT, which includes all the capabilities of the BioFlux 200 Plus, but also has the ability to work with an additional plate format, the 6-well plate. And it comes equipped with a Zeiss AxioBserver inverted microscope and Montage Plus, our image acquisition and analysis software that facilitates the automation of image acquisition and the delivery of shear flow. With its flexibility and ease of use, the BioFlux is the gold standard for shear flow research. There is no tubing to clean up after each experiment. Setup and maintenance is simple. It supports a wide range of assays, helping you generate more data faster and at lower cost. Now that you're familiar with the BioFlux, let's jump into the lab where I'll show you how to work with BioFlux plates and precisely regulate shear flow delivery to experiments. I'll also show you live imaging of Jurkat cells adhering to TNF-alpha activated endothelial cells. And finally, we'll see streamlined data acquisition and analysis using Montage Plus. Let's get started. BioFlux plates are sterile and come individually wrapped. We're going to open the packaging. And now that we don't have the package anymore, we're going to flip the plate and look at the bottom just so that you can see the viewing windows where we can find the microfluidic channels where experiments are imaged. Loading cells and reagents into the wells of BioFlux plates follows the same procedure as with a regular plate in a static assay. However, it is crucial to avoid bubbles to prevent introducing air pockets in the microfluidic channels. To do this, we just pipette our cell suspension very slowly into the wells. So we're going to load various wells on the plate. And while we're loading cells, it is always important to remember to follow proper sterile technique to prevent compromising the health of the cells for the experiment. Because we're only working with 8 flow cells, we're going to seal the rest of the plate with a plate sealer that comes included with the plate. This will prevent air from entering the microfluidic channels of the other flow cells that we're not going to use in this experiment. And this will also maintain sterile conditions in the plate. To do this, we're just going to use a razor blade to cut out the plate sealer that is on top of the flow cells that we're going to work with today. Now that the cells have been loaded into the plate, we're going to place the plate on the microscope and attach the gas interface that will deliver air pressure into the plate to create the desired shear stress rate within the microfluidic channels. In this case, we're going to flow the cell suspension that we just loaded into the plate to the microfluidic channels in the viewing windows. In our lab, we have a Bioflux 1000Z that is equipped with a Zeiss axio-observer and inverted microscope that has a motorized stage. With the motorized stage, image acquisition through the various viewing windows is automated. And now we will proceed with a live demo. For the in-lab portion of the demo, we've prepped two low-shear 48-well Bioflux plates. We've placed our first plate on the microscope and are viewing it live using our imaging software, Montage Plus. We're adjusting the lamp exposure so that you can see one viewing window with two microfluidic flow cells, one at the top and one at the bottom, each seated with human umbilical vein endothelial cells or HUVECs. Montage Plus uses the slidebook platform that is powered by 3i to integrate the operation of the microscope and the Bioflux to automate image acquisition, analysis, and shear flow delivery, all within one software package. As we move along the x-axis, we're confirming that the HUVECs look healthy and that they have grown consistently across the length of both channels. To seed the plates, we coated the microfluidic channels with 100 micrograms per ml fibronectin, then seeded the cells a day in advance to let them attach and form a confluent monolayer. Each microfluidic flow cell was loaded with 50 microliters of a cell suspension with a concentration of 10 million cells per ml. To seed a full plate, you typically need 10 to 20 million cells per ml. If confluency isn't reached overnight, you can feed the cells every day with fresh media until they reach the desired confluency. In this experiment, we're comparing two conditions. The top channel contains unactivated HUVECs, or negative control, and the bottom channel contains activated HUVECs with TNF-alpha for 4 hours to simulate inflammation. Now we'll bring the Bioflux software up and set our shear stress to 0.5 dynes per centimeter square. For reference, this system can run up to 200 dynes per centimeter square, depending on the plate type that's used. You also don't have to use the entire plate at once. You can choose specific channels to run as needed, as we have done in this case. We should soon see the Jurkat cells flowing through the microfluidic channels. We'll give them a few seconds to reach the viewing window. Because we're testing adhesion to two conditions, unactivated and activated HUVECs, we expect to see more adhesion of Jurkat cells to the bottom microfluidic channel than the top. As you have also probably noticed, we're starting our imaging in bright field, but we'll switch to fluorescence shortly, since we stained the Jurkat cells with nuke blue so you can more easily visualize where they adhere and how they flow. So here we see the Jurkat cells flowing through the channels, and you can already spot some of them sticking to the bottom channel. These appear as small, round, darker dots. We're now going to switch to fluorescence using the DAPI channel, so you can see the cells flow more clearly. Because these experiments take several minutes, I'm going to speed up the video a bit to show how Jurkat cells preferentially adhered to the inflamed endothelium on the bottom channel. When we are imaging these experiments, we normally take time-lapse images that we then use to count the Jurkat cells that have adhered to generate an object count over time with Montage Plus. I'll show you how to do this once we've gathered all our data. For now, we're going to let the Jurkat cells flow through the microfluidic channels and adhere to the TNF-alpha activated monolayer on the bottom. We can see that a lot of them have stuck already. So we're going to give the cells a few more seconds before we stop flow. And once we do, I'm going to show you how we acquire time-lapse images to increase throughput. Now we're going to bring back up the Bioflux software so that we can stop flow by simply clicking on the stop button. Since our microscope has an automated stage, the software lets us create a multi-point list where we define the X, Y, and Z coordinates of each viewing window on the plate. Once that's set up, we're going to select time-lapse, which allows us to take images at defined intervals. Here, that's going to be every minute for 10 minutes. We also select multi-point list and choose the channels we want to image in this case, DAPI and bright field. Next, we launch the BioFlux software to initiate flow in all microfluidic channels at 0.5 dynes per centimeter square. We're going to activate the delivery of pressure to every single microfluidic flow cell in the plate, and then we'll go back to Montage Plus and click Start to begin image acquisition. Now that imaging is underway, you'll see both the bright field and DAPI channels on the screen. We'll move both of these screens up so you can see them side by side. As the system captures images, the stage moves automatically between the different flow cells on the plate. In the interest of time, I'm going to speed up the video here too. After watching several of these captures, you might notice it's easier to see jerked cell adhesion in the bright field channel compared to DAPI. That's because live preview isn't available for each capture point during multi-point acquisition, but we'll be able to review the full dataset once the run is complete. So we'll give Montage Plus a few more seconds to finish up image capture, and then we'll begin data analysis. To analyze the data we just collected, I'm going to launch Montage Plus. As you can see, this software allows us to capture images through the Home tab. It allows us to also analyze the images. And it even allows us to coordinate shear flow delivery from the BioFlux system with image acquisition. This is done simply by clicking the special capture button on the home screen and selecting the BioFlux control. For the analysis portion, I'll begin by opening our experiment. Once loaded, you'll notice that Montage Plus conveniently saves all associated files, including metadata, into a single folder. To keep things moving, I'm going to focus on analyzing data from just one of the time-lapse captures. Let me start with a quick visual summary of the experiment. I'll click play so you can view a movie of the full time lapse. Now I'll pause the video and navigate to the last time point in the sequence. This frame typically shows the highest fluorescence, which makes it ideal for distinguishing signal from background noise. The first thing I'll do is change the color of my objects of interest to something that's easier on the eyes. Next, I'll open the lookup table to enhance contrast in the fluorescent signal while minimizing saturation artifacts. I'm going to adjust the bounce. A little bit, just so that I can focus on the objects or the cells that have stuck to the monolayer. And filter out some of the cells that are flowing. I'll keep it there, and then I'm going to apply this to my image. Now we'll move into segmentation. I'm going to create a mask, so I'm going to go into the Analyze tab and I'm going to create a mask to isolate my objects of interest, in this case the Jurkat cells. Like most imaging software, Montage Plus uses masks to define which elements to analyze. So once I click on the segment mask, I'm going to adjust the bounds again. And I'll start with 400. I'm going to adjust the bounds until the mask clearly outlines the Jurkat cells. You'll see this represented as a blue contour or a halo around the cells. So that looks pretty good. Now I'm going to move to different frames in the time lapse and apply my mask there just to ensure that my mask is being applied to the objects of interest. That all looks pretty good to me. Once I'm happy with my bounds, I'm going to apply this segment mask to all of the images within the time lapse. It'll take a few seconds. Next, I'll define my region of interest, or ROI. To do this, I'm going to enable the bright field capture so I can see the boundaries of the microfluidic channels. I'll use those boundaries to draw a rectangle that defines the ROI. And I'm starting with the bottom channel because this is where most of the action happened. This is the one with the activated monolayer of endothelial cells. Now that I have my rectangle, I'll convert that ROI into a mask, telling the software exactly which area of the image I want to analyze. At this stage, I'm going to specify which masks to work with. In this case, it'll be the segment mask for identifying the Jurkat cells, and then the ROI mask for defining the analysis area. The ROI mask would be mask number 1 in this case. And the output of this computation is going to come out in mask 2, but I'm going to change this name to results. Something a little bit more descriptive. Now I'm ready to get the quantitative data. I'll click on Mask Statistics up here and select the parameters I want to include in the report. So I would like to capture the date, also capture the elapsed time, and then under morphometry, I would like to conduct an object count to determine how many Jurkat cells stuck to the endothelial monolayer over time. As you can see though, the software also offers several output options. You can quantify the area, you can also measure intensity, and more. Once I've configured the analysis, I simply click on Export to Excel, and the data will be generated in seconds. To confirm the export, I'll open Excel so you can see the data laid out in a spreadsheet. As you can see, we have information on the name of our experiment or the name of our file, where we captured, as well as the date. And in the data, we can see the capture date, the capture time, the elapsed time, and the object count, which were the parameters that I chose under Mask Statistics in Montage Plus. In the interest of time, I previously analyzed a set of time-lapse captures and created a graph. So let me open that in Excel under my results. So if we go down into the graph, as expected, The results show greater adhesion of Jargat cells on the TNF-alpha activated monolayer, so that would be the line in orange, compared to the control, which is the line in blue. This brings us to the end of the adhesion workflow, from acquisition to analysis. Thank you so much for attending the webinar.